We're going to return to the concept of liquid biopsies, and as you know, the blood is a mess. There are cells in there, there's DNA in there, and there's little particles called exosomes that nobody knows what to do with. And the one person who does is our next speaker, Dr. Shannon Stott, who's an assistant professor at the Mass General Cancer Center, is someone with an engineering background with a specialty in microfluidics who's going to take this on. Shannon.
Well, thank you, Daniel, very much for that introduction. As Daniel mentioned, I'm a mechanical engineer that's been working in the liquid biopsy space for the past decade or so. This is something that my laboratory is still focused on, as I was one of the co-inventors of the CTC chip, but I'm also part of a very large and I think a great team that's focused on this, and Daniel will be presenting some of the work on the CTC chip later. But today, what I wanted to present to you today was something that my laboratory has been focused on for the past 4 years or so. And one of the things that we noticed as we're in the rare cell game is that, one, it can be a little challenging sometimes to be able to process the blood immediately, as these rare cells can sometimes deteriorate. But we also wanted to be able to get something that was actually in a little bit more abundance, but could also be complementary to circulating tumor cell analysis. So what we also started to notice is that when we were looking at these cancer cells, and here's a picture, I guess there's no pointer here. Here's a picture, an Sem Micrograph that was taken by a postdoc in my laboratory, Dr. Eduardo Riategui. And this was a cancer cell that was isolated from a breast cancer patient using the CTCI chip. And as we were looking in more detail on this chip, on the cell, you could see these small particles that were budding off the surface. These particles are thought to contain RNA, DNA, proteins, cytokines, lots of important information that could be relevant for the cancer patient care. And it was about this time that Xandra Brakefield also contacted us to see if we could develop a new technology that would be focused on isolating tumor-specific exosomes. There are lots of technologies that are developing right now that isolate exosomes in bulk, but what happens is that because all cells release these tiny particles, they can sometimes mask this tumor-specific message that would be coming from the cancer cells. So what we wanted to do was develop a microfluidic platform that can take whole blood, serum, or plasma, process it through our chip, and be able to get out that tumor-specific message. Now, one of the features of this device are herringbones that are presented in a staggered pattern on the surface of the device. And what these herringbones do is they twist and turn the blood, creating chaotic flow so that all of our exosomes interact with the surface of the device. Why would we want them to interact with the surface of the device? That's because we're putting antibodies that are specific for the cancer on the surface so that we can pull out those tumor-specific exosomes. Here's just a flow visualization of one region of the device, and as you're going up in the Z-plane, you can see that type of chaotic mixing that we're creating with our microfluidic design. Once we've run through our patient sample, and sometimes we run through as little as 0.5 ml of serum, we can then isolate out the tumor-specific exosomes, and the types of analysis that we're doing are ddPCR, RNA-seq, proteomics, as well as functional studies at looking at the co-culture of these exosomes with things such as other cancer cells. So to give you a few more details about our device, there are a few unique things that we incorporate into it that I think help us maximize our tumor-specific capture. First, we've added a tunable biomaterial that was developed in my laboratory that's just a few nanometers thick. Now, this material is made out of gelatin so that it's non-fouling, and the intention of this material is that once we capture these small particles, we want to be able to release them into a small vial, so we can either do quick molecular analysis, or if we want to do any of those functional studies that I mentioned earlier. And as you can see, just in the period of 60 seconds, all of the particles that we capture are released from the device and then are collected. What we also did was incorporate some nanoparticles onto the surface so that we maximize the surface area inside the device. And we've done some clever antibody engineering so that we can also maximize the amount of exosomes, since they behave slightly different biophysically than these large cancer cells that we're used to capturing. So using some model systems, we wanted to see, did we achieve our goal of having a better functioning device in comparison to some other technologies, as well as being able to get the tumor-specific enrichment that we originally sought? And so first, comparing our microfluidic chip versus magnetic beads using the same antibodies that we selected for our cancer targets, you can see that the microfluidic chip was able to pull out more tumor-specific material than the magnetic beads, or if we just did ultracentrifugation of serum. And again, looking for that enrichment factor that we were going after, if, as input shows, we just ultracentrifuged the sample, we had very little tumor-specific enrichment. If we use an irrelevant antibody that's not specific for cancer, and which is referred to as the IgG, you can also see that there's no tumor-specific enrichment. But if we use EGFR or a cocktail of antibodies that were selected for the cancer that we were targeting, you can see that our enrichment is 100-fold or higher. Additionally, if you don't want to do molecular analysis and you just want to image the exosomes that are captured on the chip, these are exosomes that are captured from a glioblastoma patient that were labeled with a primary or secondary antibody so that you can actually visualize them on chip. And then lastly, there's a lot of interest right now as we're learning more about exosomes that size may matter. But unfortunately, a lot of technologies are size dependent in terms of their isolation. Using our approach, we don't have any pre-selection of size on the chip. So as you can see here, we can capture a wide range of sizes of exosome, anywhere down from 90 nanometers up to a full micron in size. So that we'd be truly getting anything from exosomes to microvesicles, as well as oncosomes that may be relevant for the patient care. Next, we wanted to see what happens when we run patient samples. So I'm just going to show you a glimpse of some of the data that we've been getting. These are using glioblastoma patients, and glioblastoma is a cancer that in particular is really challenging to do serial biopsies based on the location. In addition, when we've looked with circulating tumor cells in glioblastoma, we do see them there. We see them about a third of the time, but it's not as prevalent as we'd like to see. Circulating tumor DNA, some studies have shown that it's less than 10% in glioblastoma. So we're hoping that exosomes could be something that could be transformative in their care and also understanding more about this particularly deadly cancer. So here we ran 6 patients that were banked for over 5 years in a -80, and we isolated out their exosomes from both the patients as well as healthy controls. And then in collaboration with David Ting at the MGH Cancer Center, we did RNA-seq. We were able to isolate over 1,000 significantly upregulated genes in the cancer patients in comparison to our healthy donors. And we also were able to see that we were getting very tumor-specific message that was relevant to glioblastoma. Other studies have looked at EGFR-V3 in these glioblastoma patients, and we wanted to confirm that we were able to pick these up from our patients. And as you can see down here, using ddPCR, we were able to pick up the EGFR-V3 message that was completely absent in all of our healthy donors. But what was exciting is we started to delve deep into these genes that were significantly upregulated, and we started to break apart genes that were outside of classical glioblastoma genes. We're also finding high upregulation of genes that were relevant to survival, stemness, signaling, resistance, progression, angiogenesis, a huge wealth of information. But what we're also really excited in this whole to be determined, these are genes that have not been reported in the literature that we're finding highly upregulated in our patients, and we're hoping by expanding our patient cohort, we learn more about what this may mean for these patients as they're undergoing treatment. One of the things that was also intentionally done in the design of this technology was to keep it simple. We wanted this to be a very accessible, cost-effective technology, and was also built upon the backbone of the herringbone CTC chip that we had already mass-produced at Mass General. So now we have these chips in plastic that only require a syringe pump to run so that we can rapidly expand upon our patient cohort. And our long-term vision is hopefully that we can take these chips and disseminate them to cancer centers across the country so that we can learn more about what can actually be packaged in these and what this means for cancer patient treatment. So great. Thank you.